This is a webinar today about ELIXIR TraIT pilot, and that's led by Dylan Spalding, who'll be talking later on, and myself, Sanne Abeln, and I'm working at the VU University in the Netherlands. I'm an assistant professor there, and this is done as part of ELIXIR, of course. So if we first consider the context of this pilot, there are ELIXIR human data implementation studies, and there's 4 of those. So it's on the human data aspect, and actually we're one of the 4. So you see here the bottom one. That's the one we're currently going to be talking on. This was started in June 2015, so about a bit over half a year ago, and currently it will actually be running until December this year, so about a year and a half. And the thing we're trying to do is really in the field of the big data, human data, and also mostly applied to the domain of health and disease. So the things we can consider in the human data. So if we start gathering for example, clinical studies, maybe cohorts for testing a certain drug, typical things that are collected are clinical data, so measurements. You can think about the body temperature or something as simple as height, but also some very study relevant or specific variables. Images or non-high throughput diagnostics, such as staining and the amount of staining on samples, imaging, and of course, high throughput molecular profiling. And it's specifically this last one, high throughput molecular profiling in combination with the clinical data that we'll be talking about today. Now, of course, in the human data, one of the real big issues at the moment is just the massive growth and the really big size of the data. So at the moment, the DNA sequencing data is doubling every 6 to 8 months, and the lines here, like the logarithmic scale here, so this is going really, really fast. But you also see that the human data, so the blue line here, EGA, which is also what we'll be talking about today, the repository for the human data, it started later than a lot of the other sequence repositories, but you see it's growing really fast, and it almost contains the largest amount of sequences already. And well, this is doubling very fast. The human data, of course, has one other specific issue, and that's, of course, the privacy or ethical issues around the human data. So typically, what you want is restricted access for this data. And just to give you an example of what you could do with this type of data, so if you have sequencing data, you can also, or you actually also expose someone's individual risk factors for certain disease. So, for example, Michael Snyder, he put his genome and all his sequencing data online, and that immediately showed that he had a high chance on high cholesterol, higher chance for skin cancer, higher chance on diabetes, and of course, for example, employers or insurance companies could use this information, and so we need to be really, really careful about where this human data ends up. Right. So what do we actually want to do with this human data? And typically that's, in the end, trying to make people better. More specifically, typically we're looking for biomarkers within the samples that can have a diagnostic, prognostic, or a predictive value. So what we usually do is, from the patient, we take some kind of sample, then we use a molecular profiling technique on the samples. We do a lot of analysis, and then we try to get out biomarkers that have these like diagnostic or prognostic values. So within TraIT, and I'll explain more about TraIT later, but how we see this is that we have the samples, so we have the human samples, and if we now talk about the data, this comes out of raw data, out of the machines. You need some quality control. You need to actually store your raw data. And then a lot of computational processing goes on. So this is both on the cleaning of the data, but also on, for example, the calling of the data. So is any gene specifically significantly mutated? Is an expression level significantly higher or lower than in reference measurements? So you have this cleaning on the one hand in computational sense, but also typically some statistical significant calling of the events that are interesting in the data. And so you have raw data, then you have interpreted or processed data, and there's computational workflows that link those two. And this pilot is mostly about connecting these. So it's connecting the raw data that is stored somewhere, the processed data, and almost the more rich data for interpretation, and the computational workflows that link those. Right, so if we make that a bit more specific, so the aim of this pilot that we're talking about is really about linking these three concepts. So the process data in TraIT, in the TraIT workflows, we store this process data in Transmart. The raw data in TraIT, we've chosen to store most of this data either locally or at EGA for archiving, so the raw data. And then the computational workflows that actually made us go from the process data to the raw data, they're actually accessible through ELIXIR, which is a pipelining or a workflow management system where you can annotate these pipelines very well, but also rerun them. So over the next 5 minutes or so, I will explain TraIT, so the TraIT project, but also Transmart and ELIXIR in a little bit more detail. And after that, Dylan will take over and he will explain in more detail the role of the EGA in this pilot. At the very end, I will come back to some more details of and an update of how the pilot is actually going. Right. So if we now look at TraIT, I think TraIT has now been going on for about 4 years. TraIT was set up as an infrastructure project, very specifically stating that it was not with the aim of to produce research within this project. So it's really an infrastructure project for translational research. Human data also mostly in this case. And the interesting part of TraIT, and I think also what made it so interesting, so it's a Dutch national initiative that was really end-user driven. And how we call end users here is perhaps not how we typically would call the end users in the bioinformatics community, but an end user here would be typically a molecular biologist or a pathologist or maybe even a clinician. Okay, so if you look at the structure of TraIT, you see that kind of like the earlier types of data you could collect for a specific patient. You see here that there's work packages on the different types of data. So clinical data, these are these measurement data, for example, your temperature, or it could also, for example, be survival, patient survival, clinical imaging, pathology imaging, so 2 different types of imaging, biobanking, so where the samples and how the samples are stored, and experimental data. And of course, I've been mostly involved with my role as a bioinformatician in the experimental data and the computational analysis thereof. And all of this then is combined in the infrastructure. Right. I think interestingly also at TraIT when we started, it was quite clear from the start on that there's really a lot of different people playing in these kind of large initiatives. So that would be going from PI end user, which in TraIT is defined as a molecular biologist, whereas maybe in other fields we may actually define the one end user slightly different. But there's also a lot of other people involved here. Bioinformaticians, tech operators, that lab people who may want to actually keep their electronic lab journal. So a lot of roles and a lot of users here. So if you look at the data flow through TraIT, this is how the workflows have been envisioned. And so this is the level of the sample or the patient or the participant. And then you see that all these different data types are collected again. And then there's also data integration. Now, in TraIT, we've chosen some specific tools that can solve some of these individual problems for the data capture and the data integration. So, for example, we've used OpenClinica, which is very popular now in the Netherlands. It's taken up really, really rapidly since TraIT made this well available. XNAT, and so these are imaging archives. Then we have Molgenis for the sample catalog, and then for the molecular data domain, we have Galaxy, Chipster, and the phenotype database. And today I will be mostly talking about Galaxy, and then for the integration, Transmart. All right, so if we now zoom in to the work package 4, so the molecular profiling domain, what you there mostly consider is the clinical data, the data from the experiments, the processing pipelines, and then the combining of these two different data streams again in Transmart. Well, the workflow of the provenance thereof is mostly ensured by Galaxy. So if we look a little bit more in a bit more detail in these tools, this is Galaxy. So what you can do here really well is build up this processing pipeline, your computational processing pipeline. And so this is, for example, from the raw data.
I don't like to say anything because the recording.
This is, for example, from the raw data to the process data, and so here you can set your methods that you want to use for the cleaning, for the calling, and you can make these logical steps, and you can make them inputs and outputs of the different methods, and so you can make a workflow. And the nice thing here is, of course, that in a kind of visual way, this is now not only available as a bioinformatician would usually hand over this in the form of scripts, but it's also available in a visual workflow registry. And of course, you can reference these workflows and publish them as well. Transmart is really used to combine the process data, so when the data is already processed, together with the clinical data. And so this is, of course, where, from the TraIT end user point, the really interesting combining of data starts happening, because this is, for example, where you can start stratifying patients to see, for example, if there are certain patient groups. So this is a classic Kaplan-Meier plot. If, for example, certain group of patients, think, for example, about a certain mutation being present or not. And so you here see that the BRAF for the red line, if you have a mutated version, you see that the survival time here is lower for this group of patients. And so the really richness of Transmart is combining this clinical data with the processed molecular profiling data. Transmart allows some specific analysis. So this, for example, copy number gains. You see here over the different chromosomes, and you can see here over all the patients. You can see what typical profiles are for two subtypes of cancer here for patients. Another thing that we focused on in TraIT is combining samples from different patients. So this is something you may recognize as, for example, something that will be shown in CBio portal, the OncoPrint. What it really does is here per sample, per block, you can indicate for a favorite gene, for a specific gene, if there is a gain event, if there is an up or a down regulation event, if there's some specific mutation. So you can combine different molecular profiling techniques and collapse them together. And we've made quite a bit of work to make Transmart in such a way that you can actually have enough information to be able to build these kind of plots. Right, so here I will actually hand over to Dylan, who will be introducing EGA in more detail.
Yeah, so I'm Dylan. I'm project manager at the EGA at the EBI. Thank you, Sanne. I'm going to talk about the European Genome-phenome Archive. Now, this is a joint service for the European Bioinformatics Institute and the Centre of Genomic Regulation in Spain. We're a secure and permanent archive for all types of genetic, omics, and phenotypic data for humans. Now, all this data is consented for research use, and I'll talk about that on the next slide. The data access is managed through application, and all data is encrypted, both storage and in delivery. And our submissions include the raw data from genome sequence experiments, transcriptome, epigenome, and proteomics experiments, and we also take process data such as covariants and genotypes and sample phenotypes. So what is controlled access data? Well, you've probably heard of some of these open archives, such as ENA for sequence data, ArrayExpress for array data, EVA, biosamples, and so on. But EGA is for controlled access, so it will take all these forms of data and control the access to them depending on who's been granted access. So the data we get has to be informed consent by a patient or a member of research to reduce their genotype data, identifiable data, to certain people. So this data is all human, and it has personally identifiable data in it, such as raw data, which is BAM files, CRAN files. We can have process data, and we can have phenotypic data. This data is generally affiliated to biomedical research or consortium projects, for example, TraIT, and access is by formal application procedure to a data access committee, or DAC. So some context and metrics about the EGA. It was launched in 2008 as a secure archive for controlled distribution of consented data. We have around 7,500 users who've been granted access to one or more data sets. We have submission accounts for over 360 users. The total size of the archive is over 3.3 petabytes, and that's divided into approximately 1,800 data sets. And we've distributed over the last year 1.6 petabytes of data. We have a help desk, which has around 200 contacts per month. And some of the example data that we store are Wellcome Trust Case Control Consortium, genome-wide association studies, and cancer data. So this is an illustration of the data growth since 2010, and as Sanne was mentioning earlier, there's been a massive growth in genomic data that's been archived. And in terms of distribution, we've been distributing data, and this has been increasing quite a lot as we've developed a streaming downloader in the last year or so, which I'll talk about later, which integrates with Galaxy. So in terms of some of the studies at the EGA, the majority of the data is actually cancer data, which you can see here. But there's a lot of other disease and phenotypic data here. Noticeably, we have a lot of exome sequencing data. That's probably due to the high coverage it gives. And we've got array-based data and whole genome sequencing data as well. These are some of the consortia that have archived at the EGA. There's TraIT, but there's also Cancer Research, the Wellcome Trust, UK 10K, RD Connect, and so on. So how do you access data at the EGA? Well, the data at the EGA is stored in studies. And for example, here's a screenshot of a study page for the UK 10K data. Each study can have one or more data sets associated with it. So for example, here you can click on one of these data sets and you'd end up at the data set page. Now, it's the data set that a user can be granted access to or not. So here the screenshot shows someone who's come in browsing. They can find out a certain amount of metadata, i.e. the data set, which study it belongs to, the number of samples, but they can't actually access the data at all. To access the data, they have to apply to the Data Access Committee. And the links there are down on the bottom right-hand side. So here, if we start at the left-hand side at the bottom, we have a user. He comes to the EGA website, and he decides he wants to access, for example, Malaria Gen or Wellcome Trust Case Control Consortium data. So he then applies directly to them, the DAC, and not to the EGA. That DAC will grant or deny access, depending on their operating procedures. If they grant access, they will directly create an account at the EGA, and that account then will be tied to that user. Once that user has an account, they can log into the EGA using that account and then download the data sets that they've been granted access to. As an example, this is for the Wellcome Trust, and this is the form that you have to sign to gain access to their case control data, which basically means you mustn't try and identify the individuals or share the data, and you must have research use for it. While you can discover data at the EGA using keywords, for example, the technology used or the disease phenotype and so on, we also want to be able to increase access by determining whether, for example, a particular variant has been seen within a data set, and then you may be interested in that. And for that, we've implemented a beacon, which, as you saw earlier, was another of the human data implementation study projects from ELIXIR. And here we have the UK 10K project. Someone's queried it, and they've seen that there is actually a variant somewhere in the UK 10K project of chromosome 4 at position 260998, I think it is. So if you're interested in that variant, you can now then apply for access to that data set and then do your own analysis on it. So here's an example of the life cycle for data within the EGA. At the bottom, we have the anonymous user who comes in and browses the EGA website. If they find something interesting through Beacon or just by keyword search, they become what we call a requester. They apply for data access by the Data Access Committee, who then creates an account if they grant access. At that point, they can then download from the secure encrypted archive the data in encrypted form. If we add a submitter to this, then the submitter can upload data via the EGA encryptor, which encrypts the data from the submitter's computer and uploads it via STP or Aspira to a Dropbox. And they can also submit the metadata via Webin or REST, which I'll talk about in a minute. Once the metadata has been submitted, the file data in the secured inboxes is then processed into the archive. And once the study or data set has been released, it's then available for download. So for submission, we support primarily two types of submission: programmatic submissions via a REST interface. This is where you describe the metadata using XML. And for example, you would describe a study using an XML, the dataset using an XML, and samples, experiments, analyses, and so on. So all the metadata would be in individual XML files, which you would submit via REST. Alternatively, you can submit via a graphical web interface, which we call Webin, which will take you through a complete submission step by step in a graphical environment. So for accessing EGA data, there are basically three steps. You request the data to download. Well, once you've got an account, you request your data to download. You specify the data you want to download. You specify an encryption key to use for that requested data. And then the downloader will create a URL, one for each file that's in the data set. You'll then download that data and stream the data from the EGA archive to the user storage via HTTP, or you can data stream the encrypted stream with a key specified in the request. Once you get the data, you can decrypt the data to start using it, or we've now started to develop a fuse layer which actually allows you to directly interact with encrypted data and saves you having to decrypt it. So overall, I've described the EGA and the way it fits in with submissions and output. And so the downloader that I've just been talking about can directly integrate with the Galaxy pipeline that Sanne was talking about, and the Transmart can actually submit data via the REST interface, so it becomes fully automated. We can also pull these IDs from the EGA back into Transmart and Galaxy to link the raw data in EGA to process data in Transmart and the processing within the Galaxy pipeline. That's all I've got to say, so I'm going to pass it back to Sanne to complete the presentation.
Can you hear me now?
Okay.
Yes.
Okay, good. I'll go on. Okay, so this is exactly what Dylan just showed, the aim. So we're linking the process data, the raw data, and the computational workflows. And actually, now I wanted to look at a little bit more detail of how we're doing that and at what level, because this is a pilot, so we're only kind of making a proof of principle here for a showcase, but also at the same time we make sure that it is a more generalizable framework that is actually maybe even going beyond the tools that we're currently trying to use. So back to the TraIT picture. So you have the human samples with the clinical data and experiment data. You have the processing workflows. You may have different workflows depending on the actual type of experimental data, and for a single sample you may actually have also multiple analysis, of course. Now in Transmart, all of this is combined again. And this is then where the processed and the finalized clinical and process data will reside, and this is also heavily curated. So once in Transmart, and this is the TraIT way of doing things, it will be a curated data set. So Transmart is for the storage of curated data. Now, the experimental data, the raw experimental data, does not go to Transmart. So there we either need to store it locally, but for archiving purposes it's of course way better to be able to put it in EGA. And actually all the projects at the moment I think that are supported by TraIT are actually human data. So everything actually needs this controlled access archiving. So now of course what we really would like is kind of linking these things back. And the one other thing that comes here is the workflow provenance of these computational pipelines, which we already saw earlier, can be built and also set up in Galaxy, and at the same time that ensures that you actually remember and know how we went from this raw data to this process data. Now the first thing you would really like to do is something very simple, and that is once you're at the process data, you would actually also, rather than only knowing what's in there, you would actually like to be able to know where your sample is, but also where your raw data is, so where the storage here. So one of the simplest things, and this is, we've been looking at this in the first half of the thing, how can we reference in Transmark back to EGA? And of course, to start doing that, we first needed to upload some data also to EGA to get this done. Now the second part, and that's what we're currently working on, is once you know where your data is, so you know where it is in EGA, can we now actually use it again to restart our processing workflows in Galaxy? So can we take it as the first step of Galaxy, actually retrieve the raw data to start the rest of the workflow analysis? And at the moment, Juri Hoogstraat at Erasmus has been building this, and he has now a first prototype of this retrieving data in Galaxy, but also running the rest of the pipeline afterwards, working for a specific type of data. So this shows us in principle this can be done, and of course if you put a Galaxy on a secure server, then this whole process can be done securely as well, which is important. Now the last thing, of course, what you would like to do is actually also in Transmart go back to the reference of the workflows and actually be able to automate this process. Now to completely fully automate this is probably beyond the scope of this pilot, but you should at least be able to make this logical loop for an end user. So the idea here is that once you're working in Transmart on some specific samples, you see maybe that you make a stratification on different patients, and you see, for example, that one specific patient, you expect a certain mutation there, but in Transmart, in the process data, at least it's not been seen as significant. You may actually want to go back to the evidence for that data. Maybe the region wasn't sequenced properly, or maybe it was just below the significance level. So you may want to actually rerun and inspect the raw data in Galaxy and rerunning part of your processing workflows. So to build all of this, we actually use the TraIT cell line use case, which has a lot of different types of data. So we have DNA data, we have RNA sequencing, we have different types of arrays, we have copy number data, miRNA data, and also proteomics data. Now this is not privacy sensitive data, and this means that it's very good for testing and developing workflows when they're not yet secure. And it's also very useful for demo and training. So once we want to actually show the technology we have built, we can actually use this to also let people play around with the systems. if you now look at the full kind of workflow I explained to you earlier, how this would look is that an end user, maybe someone who's not really a bioinformatician, but someone who is actually is really interested in the study that person is looking at, is exploring a data set in Transmart, may make a certain patient stratification, and may become interested in one or two samples that behave differently from the rest. You can actually already see the cold data, but at some point you may want to go actually back to the raw data. At that point, you want to know where the raw data for the specific samples are and also how they were processed. And the idea is then to, in Transmart, build a link where you end up with showing where the raw data is, and then either having a link and downloading it, or copy and pasting this link, perhaps in a more simplified version, into Galaxy, where you can now upload the data from EGA, so with this importer, and actually hang it on as an input set to the rest of your pipeline, so you can rerun the entire pipeline. And so the output would be the same as before, or you can actually try and change some parameters. On top of that, in Galaxy for TraIT, we've also got some specific, more dedicated analysis tools that also need to have the more raw versions of the data to actually be able to create the analysis. So also this would now be accessible. So if you look at the full picture then, you've kind of made this round, and this part that is still indicated here in green, where in the end you may actually also want some clinical parameters to use in your workflow analysis, is something that still needs to be worked on. But in principle, you should be able to make this full loop using the reference data and the retrieving of the data of Galaxy. Now, of course, there's other ways you could set up the system. So, for example, for a lot of touch studies, storage of the raw data is actually in two places. So typically there's local storage and archival storage. And sometimes that local storage could actually be very close to your Galaxy server of interest. So you might not actually want to get it from EGA, you may actually want to get it from a different position. At the same time, there's different ways of looking at your process samples. So we have something like the sample catalog, but you could also, for example, something like cBioportal. Also from here, you may actually want to have a reference back to your data and back to your workflows. So another part that we've done in the first part of this pilot is actually try and create an ontology that actually makes this linking between studies, patients, and actually data sets or process data available, and being able to link the locations of the data and the EDA locations to the specific concept. So this is really also a mapping between the raw and the process data. And if we have a more generalized framework here to be able to do this, that also means that we can use possibly different tools and kind of connect them in the end in a similar way. Right, so the timelines for this pilot. So the first part has been running from June to December. We've worked very hard on the upload and the download of the test data, and the first part of the cell line use case is now happily in EGA. We had a first draft of a more extendable metadata scheme. Then currently Yuri is working on this Galaxy import from EGA, and at the Hive, the continued upload of the cell line use case data for TraIT is ongoing, and the upload to EGA we're talking about here. Then in the very last part, we actually want to go back to the first part and make this a full working pilot, where you really can go from Transmart to Galaxy and then retrieve the raw data and start a workflow. And of course, from the EGA perspective, we have, during this process, we've had continuing work or support from the EGA, both from the CRG and the EBI. All right. Now, one of the things that we also hope that this pilot is leading to is actually trying to make the data more fair in the way that it's findable, accessible, interoperable, as defined nicely in this recent publication. And so it's quite clear that with huge numbers of data, but especially with the very big data, so we have a lot of data on genes, the number of samples or the number of patients in these studies are still typically relatively small. So that means like actually sharing of data becomes more and more important to be able to find smaller effects. That means you need to go up to a more interoperable data framework, less study-centric, and more sharing. And this, of course, really helps in the first step to also make controlled access data more fair. Beyond the pilot, so there's already been a lot of interest in this pilot, both from the Transmart EBA connection to the Galaxy Transmart connection and the Galaxy EGA connection. So, for example, the rare disease use case in ELIXIR, but also in RD Connect, is really interested in this work, BBMRI, matching this in a similar way to sample catalogs to EGA raw data. And of course the push to fair data. Right. So in this pilot, 3 major ELIXIR parties have been involved. So this is the EMBL-EBI and mostly the EGA. The Netherlands, which also has, of course, a large role in interoperability, and so that's also one of the reasons why this quite naturally falls in there. And of course, there's the TraIT project in the Netherlands. And then, of course, ELIXIR Spain, which at the CRG also has the EGA part. Then, of course, I would like to thank the entire EGA team for all their support, and the people specifically been running this pilot. So Dylan, Jordi, earlier on Justin, Susanna has been great in helping out and getting this back on rails. Siko or Chao Zhang has been working at the VU on this pilot. Juri Hoogstraat at the EMC, Jochem first at the VU, now at together with Wibo at the Hive, uploading the data from TraIT to the EGA. And therewith, I want to say thank you. And then one last remember, we're also currently organizing ECCB, or at least in the Netherlands, which will be in The Hague in September. And it's really, so the ELIXIR track submission deadline has just closed, and there's over 50 submissions, so it's really interesting to see that there's such a big interest in ongoing ELIXIR efforts and services.